false utopia of one unifying description of the vulnerable atherosclerotic plaque. A call for recalibration that appreciates the diversity of mechanisms leading to atherosclerotic disease. A presentation from Gerard Pasterkamp, UMC Utrecht. Published as brief review in ATVB in 2022. This video is narrated by a AI-generated voice. Atherosclerosis is a complex disease characterized by the formation of arterial plaques, with a broad diversity of morphological phenotypic presentations. Researchers often apply one description of the vulnerable plaque as a gold standard in preclinical and clinical research that could be applied as a surrogate measure of a successful therapeutic intervention. The plaque that is lipid-rich with a thin fibrous cap and many inflammatory cells. Pathology studies already revealed that also more fibrous plaque types can lead to a clinical event, such as myocardial infarction and stroke. For example, plaque erosion without rupture is frequently observed and seems more common in younger females. The classical description of the vulnerable plaque is subsequently used as most important measure in search for genes, proteins and cell characteristics that may destabilize atherosclerotic lesions. In addition, the traditional description is used to interpret image characteristics of human lesions and of phenotypic changes in the plaques observed in atherosclerotic genetically modified mice. If a study shows a gene-specific effect on lesion phenotypes in mice, then these outcomes are also often translated to human disease by assessment of RNA and/or protein expression in human specimen that fulfill the classical description of the vulnerable plaque, the gold standard. We have to realize, however, that the most widely used description of the vulnerable plaque originates from observations of the worst patients, those who died. Studies now also show that the plaque characteristics have changed over time, due to improved therapeutic strategies and changes in lifestyle. The increasing knowledge generated by omics studies of human atherosclerotic plaques will fine-tune the definition of the arterial plaque at risk for a thrombotic occlusion. The cell-specific characterization of gene expression levels will provide insights in the cells expressing genes that have been identified in coronary artery disease genome-wide association studies and novel cell clusters that could point to mechanisms of disease progression, such as cellular plasticity, clonal expansion and ligand receptor interactions. We foresee the following future perspective. A manifestation of ischemic adverse events is recognized by many underlying mechanisms and risk factors resulting in a broad diversity of atherosclerotic plaque phenotypes. B. Next to a pathology-based description, future plaque phenotyping can be executed including bulk transcriptomics and proteomics as well as single-cell analyses. These analyses need to be executed in a sex-stratified manner. C. The analyses result in the appreciation of a diversity of lesion types that will ask for stratification by clustering. D. The novel combined pathology and omics-based plaque clusters will influence the interpretation of experimental outcomes in mouse models, and will ask for a search of the best stratified matches between mouse and men. The landscape of the atherosclerotic disease and the plaque phenotypes are complex and diverse. The natural history of atherosclerotic disease cannot be captured in one book that describes a single unifying concept. The definition of the plaque at risk can be fine-tuned if data from large omics studies are included on top of pathological characterization. Do you want RO read this review? It has been published in Atherosclerosis Thrombosis and Vascular Biology in 2022.